California. The title of his research is Time-Lapse 3D Telomere Dynamics, Modeling Properties and Quantitative Evaluation.
Jesus. What did it say?
This isn't a computer, it's my wife. Get off. Like, mom's pretty weird. Yeah, I really want this.
So hi everyone, my name is Jang and my project is on modeling properties and quantitative evaluation of time-lapse telomere dynamics. So before I talk about my project, I want to give a brief background on what telomeres are. Telomeres are basically repetitive DNA sequences found at the ends of chromosomes, and due to the end replication effect, every time a cell divides, the telomeres get a little bit shorter. So you can kind of think of telomeres as a fuse. The cell divides and ages, and eventually when the telomeres are too short, the cell stops dividing and goes to growth arrest. Now as it turns out, telomeres are very important in the study of cancer, aging, and chromosomal stability. And for my project, I'll be focusing on the cancer aspect. So it's been shown that telomere organization actually plays a huge role in carcinogenesis. Cancerous telomeres exhibit more pancake-like distributions within the nucleus, as well as the tendency to form aggregates and more telomere jittering and colocalization with certain proteins. So a study in 2007 showed that more telomere clustering is present in cancerous cells. However, looking at telomeres under a microscope is not very effective because two-dimensional images don't present an accurate portrayal of what's going on inside the nucleus. For example, here I have an image of a healthy cell, and the telomeres have been tagged with fluorescent proteins, and a cancerous cell. And now this is very useful because they practically look the same. I can switch these around and probably wouldn't be able to tell the difference. So simply put, two-dimensional telomere imaging simply isn't tangible at all. Now the study in 2007 did look at three-dimensional telomere dynamics, but unfortunately, modeling that is time-consuming, and when considering the amount of data presented in the study, it would be very tedious to model this over time. So for my project, I wanted to figure out a new way to look at three-dimensional telomere dynamics and visualize and quantitate these telomere dynamics in an intuitive way. So for my project, the Guarini lab provided confocal microscopy data of 3 cell lines. There's 3T3, which is a standard mouse fibroblast, and they're the healthy cells, but they're immortal because they're stem cells. I'm also using U2S, which is a human osteosarcoma, or a bone cancer, and as well as HeLa, which is a very famous cervical cancer cell line. Now, the images are basically 100 time sequences each, and each time sequence is comprised of a stack of two-dimensional images, forming sort of a three-dimensional model. Now, some of you might be wondering why I have a fat rabbit here as part of my slide, and it's because it'll be the solution to the telomere visualization problem. So this is actually made using Blender 3D, and it's an open source free hobbyist animation tool. I'm actually kidding about the rabbits. But Blender is useful because it's extendable through Python. So I'll be writing a script to facilitate the visualization and quantification of time-lapse telomere dynamics. So, what do you get when you combine over 10,000 microscopy images with fat CGI rabbit technology? A fully automated way to visualize telomere dynamics over time and measure them. So here we can already see some structural differences in the telomere organization between cancer cells and healthy cells. And the green little spheres are telomeres, and the lines that bound them are a kind of polyhedron called convex hulls, which is basically the smallest polytope that can be enclosed around the point. So as you can see, some of the telomeres represent the vertices of this hull, and others are actually inside of this hull. So now consider this. Modeling the same amount of data presented in this study, let's say would take an hour or so to construct this model and the telomeres, provided that you somehow got the telomere coordinates from the images. For the same equivalent amount of data presented in the study, that's over 12.5 days of constant work. No eating, no sleeping, and probably unhealthy. So here's a bounding ellipsoid model which describes the same telomere parameters, and this is just a sample demo animation. But it describes the telomere organization in a more similar fashion, similar to the 2000 study that figured that telomere organization is more oblate in cancer cells. So this allows us to quantify roundness values as well. Okay, methods. I'm not going to try to bore you guys too much with the algorithmic technicalities too much, so I'll just talk about the basic methods that I use to create these models and measure them. So the single cell GFP tagged telomeres were provided in confocal microscopy images by the Guarini lab, and there were 100 frames over 35 minutes. Each frame is basically a stack of 35 two-dimensional images. So the first step is to find out where these telomeres are. So a threshold was adaptively determined by finding 3 standard deviations of Gaussian noise added to the minimal mean of the noise levels within the corners of each model. And based on this threshold, if a point has a larger intensity than the threshold, it's labeled as a white telomere. If not, it's labeled as background. So as you can see, a lot of the noise has been removed by this binarization process. The next step is important because not all of the noise is eliminated by this binarization. So conventional two-dimensional smoothing algorithms are inefficient for 3D data because they result in overshrinkage of the top of the spheres. So I devised a 3D structural element to be rolled around the inside and outside of these telomeres, figuratively speaking, to get rid of a lot of the pepper and salt noise. So after the telomeres have been denoised, I implemented a 3-dimensional flood fill algorithm, which basically finds out which points are the telomeres. So any white telomere point and all adjacent white points are labeled as a telomere, and the coordinates are exported as a text file. So I implemented all of the above algorithms together in the Visual Studio C++ environment, and for lack of a better name, I call it the telomere analyzer tool. So next big question, how do we display this data? 3D modeling is a subject that's been barely explored in the field of molecular biology, and much less in the field of telomere dynamics. So instead of having to write my own rendering engine to look at this data, I took advantage of Blender's Python extendability, and basically I wrote a script that imports these coordinates and generates the animation. So here you can see a snapshot of Blender's interface. And it can both generate the convex hull models by calculating and measuring the parameters, as well as an ellipsoid model. So you can see I'm not very creative with naming my software, but telomere visualization. Results. So to validate the segmentation error of the telomere analyzer tool, I plugged in a phantom data set and found out using root sum of squares error how accurate these telomere points were, and I found out that when signal to noise ratio is greater than 16.5, segmentation error is less than 4/50ths of a micron. So 4/50ths of a micron is pretty accurate. Here are the graphs displaying convex hull parameters changing over time. As you can see, there's some interesting consistencies in certain parameters such as the 3T3 volume and surface area, as well as the U2S roundness. And I'll visit back to roundness later. Here's some other significant points by looking at the mean data values. And I found out that the HeLa cells have the largest volume, and the U2S has the largest surface area, which isn't surprising because the roundness is so large, as indicated by a very oblate shape. And wait, there's more. HeLa cells demonstrated the greatest data fluctuation in every single parameter. And note that it's a cancer cell. So here I've shown a new novel visualization and quantification algorithm to look at telomere dynamics. And although I've showed some interesting comparisons between cancerous and healthy cells, this project has way more applications. For example, we could implement a heuristic algorithm to a database. So a user would upload their microscopy images to this database. It would be automatically segmented, and based on comparing these memory economic models to previous models and looking at patterns in convex hull changes over time, we can diagnose cancer. Another application of this project is to look at drug therapy. Anti-telomerase drugs have been really popular over the last decade, but it's important to realize that some of these drugs might have adverse effects on the actual telomere organization. So by using this algorithm, we can figure out whether these drugs are having adverse effects on telomere organization within the cell. So in this study, we demonstrated a novel way to visualize telomere dynamics and quantify them over time. And this has huge applications in cancer therapeutics, hopefully paving the way for better treatment therapy in the future. Thank you.
At one point, I think you might have answered it some, but you said there was a different geometry in the cancer cells than in the normal cells. And also, would you comment on what led you, how'd you come by this, to do this? What was your inspiration?
So the question is, how did I decide to do this project?
That and just the comment on the geometry differences.
Okay. So I'll answer one question at a time. Regarding the geometry of the cancer cells, I did validate that the roundness values in cancer cells are particularly oblate. In fact, the lab that I got this data from, they had a previous study showing that cancer cells are particularly oblate in telomere organization, meaning that the telomeres are organized in more of a pancake distribution. I've also noticed that the telomere organization is very, very fluctuating in HeLa cells, which are known for their high chromosomal instability. So it did validate some interesting properties of these cancer cells. Regarding your other question on how I decided to do this project, so I've done a couple of years of science fair projects already on telomeres, and this year I wanted to look at a more computational modeling project because I'm a Blender fan. I was wondering how I could kind of creatively integrate Blender into my project. So I figured that since there's basically no research done on modeling telomeres, and since 3D applications are really important, this would be a cool project.
Thank you, Eric. I have a question, which is, you said that for each one of these cell types you had a time series stack of photos. I think it was 100 samples, 100 time samples. How were those manifested? Like, those weren't manifested in the samples you showed?
I'm sorry. So let me try to ask if this is a later question. So to clarify the kind of data that I used 3 cell lines, and for each cell line there was 9 samples, and per sample there was 35 minutes worth of images taken over 100 different sequences of confocal microscopy, and each confocal microscopy sequence is 35 stacks of 2-dimensional images.
So that's that 35 minutes. How did you visualize that in Blender? That wasn't obvious in the presentation.
So the animation is done over time. So the camera is basically revolving over a dynamically changing model, and basically every time the keyframe changes, it displays another time point in the animation, which is representing 35 minutes. So, take this one. Yeah. So as it's changing over time, it's representing different times in the confocal microscopy.
So, okay, when I watched that for the first time, I thought I was just observing the rotation.
But it's actually changing over time.
That's actually cell function.
Yes.
Other judges' questions? Audience?